These are notes on the cell cycle and mitosis, and you can find more information on pages 578 to 580 in your textbook, if you borrowed one. So this is a cell cycle overview. In general biology, which is what we're taking right now, we spend most of our time here in mitosis. What mitosis is when one cell splits into two. And so that's where we're going to focus the majority of our effort. But as you can see, based on this entire cycle here, actually the cell spends a lot more time in this phase called interphase than it does in the little bit of the splitting. But especially when we're talking about development of cells and cell differentiation, obviously talking about mitosis makes sense. But I do want you to be aware that the majority of a cell's life is spent in this thing called interphase. So I'm going to go over that pretty quickly. I am really not as interested in you knowing the specific names of each phase and each part of the cell cycle as I am in you knowing kind of like just what's going on in general. So interphase is divided into 3 or 4 stages, kind of depending on the cell. So there are these G stages. There's G1, G0, and G2. And G in these cases stands for gap. And again, if you remember that, fantastic. If you don't, I'm not certainly going to stress about it. So we're going to skip G0 for a moment. We'll get to G0 in just a minute. But gap 1 is where the cell is growing. So as you can imagine, if we now think about what we know of conservation of mass, we know that if one cell divides into 2, those 2 cells are clearly going to be smaller than the original cell. I mean, together they'll be the same size, but right, if you have one cell and you divide it into 2, you now have 2 smaller cells. So at some point the cell has to grow to get back to its normal size. So that's what happens in gap 1, and that's also where the organelles double themselves and make more. Because again, if you have one cell with, let's say, 50 mitochondria, when it divides in 2, I mean, it's probably not going to divide exactly 25 and 25, but you get the idea. There's going to be somewhere around 25 mitochondria. So now that cell's got to grow and make more mitochondria. So that's what's happening in gap 1. Then in in the S phase, S here stands for synthesis, and let me try to write out synthesis for you. Alright, there we go. So now I have properly written synthesis for you without you having to sit there and watch me write it. Synthesis we've heard a lot. We've heard photosynthesis, we've heard biosynthesis. So synthesis means essentially making. Biosynthesis is, making more structure, for example. So synthesis in the S phase is where the DNA replication occurs. So obviously you have a certain amount of DNA, and we'll get into that in later units, but if you're going to make 2 cells, you need to replicate all of that DNA. And then the cell goes into what's called G2. So that's gap 2, and that's where the cell is really kind of doing its last minute things before it starts to divide. There's also this funky G0 over here. So G0 is essentially where the cell is not doing stuff to prepare to divide. It's just hanging out, being a cell, doing its stuff. And there are some cells that spend maybe a year in G0, maybe a couple of weeks. It depends on the cell. Later this unit, some of you will read an article on the age of different cells in your body. Some cells are in G0 indefinitely. They never divide. So like the cells of your eyeballs, with the exception of your corneas, cornea cells do undergo mitosis, but the rest of your eyeball cells don't, which is why like babies look like they have such big eyes, or you often can't see the whites of their eyes because the little eye openings will grow bigger, but the eyeball doesn't. So if you've ever looked at a newborn baby, they seem to be like all iris and pupil, without a lot of whites of their eyes. And then as they grow, the, slits in your face, whatever you call them, the eye openings, grow bigger, and then you can see more of the whole eyeball. Some of your brain cells are just in G0 for your entire life, which is also why if you sustain brain damage, either through concussions or through drug use, or really those are the two main ways we sustain brain damage, some of that's irreversible because those cells never divide and make more. Other cells divide quite quickly. Skin cells divide, boy, I don't know. I'm going to make up something and say like every 2 weeks. It could be more or less than that, but I know it's pretty quickly. So it does depend on the cell, but that's what G0 is. It's for cells that have kind of left the cell cycle and are just doing their thing being cells. And then we get, once that passes G0, into mitosis, and we're going to focus on that with a little more detail. I do want to say that there are some checkpoints. So checkpoints are your cell's way of making sure that everything is going the way it's supposed to. So there's a checkpoint here, there's a checkpoint here, and there's a checkpoint there. And those checkpoints are ensuring that the DNA has replicated properly, that the chromosomes, that's what the DNA, a chromosome is like one big long length of DNA. Again, we'll get into some of these details a little later. But that the chromosomes have formed properly, that the cell has enough reserve energy, so often the cell uses a lot of proteins to replicate. We'll talk about why again later. But that sort of thing. And if the cell finds that it is lacking in any way, instead of an 8th grade student who will simply turn in an insufficient lab, the cell will stop what it's doing and try to fix the problem. And it actually has one of two solutions. One is to just stop and try to remedy the problem. So if it's not big enough, if all the organelles haven't doubled, things like that, then it can fix that. There are times when the cell is just too far gone to repair, and then the cell will literally blow itself up rather than continue with a faulty set of issues. One thing that is interesting to some people is where cancers come from is that those checkpoints aren't working. So it allows the cells to just divide out of control. And that's what a tumor is really. It's just an overabundance of cells. And so that's the main issue with cancerous cells, is that those checkpoints have essentially been disabled. So then where we're going to focus, because this is general biology, is on this M phase. M stands for mitosis, and really it's mitosis and cytokinesis. I don't care if you want to remember all of this as part of mitosis, that's fine. Again, I'm not real hung up on you understanding or on you remembering the specific names, as long as you understand kind of the process of what's going on. So we're going to look at mitosis in more detail, but I do just want to review before we get there that what the cell is doing before it gets to mitosis is growing, maybe just being a cell for a little while, doing what it's supposed to do, replicating the DNA, and then getting prepared to divide. Alright, so we're going to start with prophase. That's the first stage. And one thing to note is these stages blend into one another. So later this unit we're going to look at some slides that have onion roots undergoing mitosis, and you'll say, well, I can't tell, is this prophase or metaphase? Well, it might actually be what's called prometaphase, which is kind of between the two. I like to think of mitosis as a dance of the chromosomes. And so if you think about how one thing flows into the other, that will help you understand better. I like to give you the names because for some of you that helps you categorize, but then in class I'm actually going to show you, I kind of use my hands to show how the whole process works. So on each slide I've given you kind of a drawing of what's happening at that phase, and then also what it would look like in a real cell, a photograph if you will. So in prophase, remember the DNA has already replicated And what it does is condenses into chromosomes. So DNA, when it's in your cells, actually isn't in these nice, neat packages. It's sort of all jumbled up. Imagine you've thrown like some earbuds and a charger and maybe some other cords into your bag. As you go along throughout your day, they tend to just get jumbled by themselves. All that movement jumbles them up. Well, your cells are always moving too as you're moving around. And so the DNA gets kind of jumbled up. It is organized a little bit, and we'll talk about how it's organized later, but this idea of chromosomes, most of the time the DNA is not in chromosomes. So the first thing that needs to happen is, now that it's replicated, it needs to actually become chromosomes. And you can see those are these string-like things. So those are just really long stretches of DNA. And then also the nuclear membrane starts to disappear. So some of you remember from your organelle project that the nucleus is a membrane-bound organelle, meaning it has its own membrane. But in order to divide, that membrane's got to disappear. It's a little hard to see in a picture other than to kind of go, oh, it kind of looks like it's here, there, and maybe not there. Obviously, in a drawing, it's a lot easier to see. And then we have this organelle here called a centrosome. And again, remember it or don't remember it, I don't care. But these centrosomes start producing spindle fibers. And these spindle fibers are going to be really important to move the chromosomes where we need them to go. I think one or two of you did centrosomes in your organelle project, and you noted that normally there's only two of them. So this one and this one, they're at right angles to one another. But remember, all the organelles double to prepare for mitosis. So right before mitosis, you'll have a second centrosome. And then once the cell splits, each cell will each only have one centrosome again. Alright, so then in metaphase, the chromosomes all line up along what's called the metaphase plate, which is really an imaginary line inside the cell. It's not an actual thing. But what you have here are your centrosomes at either side. This is going to screw some people up because on this it says centrioles. The two parts of the centrosome are called two centrioles. Again, do I care if you remember the name? No, not at all. But you'll notice they're making these microfilaments, these spindle fibers. And again, that's what's going to cause the movement of the chromosomes. This is actually two chromosomes here. And so if you remember, all the DNA is replicated, so there's an exact copy of every chromosome in the cell. The reason for that being, then when it splits, each cell will get one chromosome, one copy of each chromosome. And so what they do is they line up with their identical copy. These are called sister chromosomes. If you remember them, awesome. If you don't worry about it. But the idea is the DNA is replicated. It's got to line up with its exact match so that when the chromosomes get pulled apart, one of each pair goes to one side and the other goes to the other side. So you can see in here, they're all lining up along this metaphase plate. And again, you'll be able to see this in some of the slides we look at. Then we have anaphase. In anaphase, the chromosomes break apart at the centromeres. The centromeres are just a fancy word for saying where they were connected before. And you can see they're being pulled away from each other, but each side is pulling away one of the two halves. And so this side is getting its 3 red ones, this side it's getting its 3 blue ones, and so each side will have 3 red and 3 blue so that they're exact. And so now what's happening is those spindle fibers are pulling those chromosomes apart. And so the sister chromatids, or the two chromosomes that are equal, migrate to opposite sides of the cell. So they've all lined up together, and now they're being pulled apart so that each side of the cell will have one of each chromosome. And you can see that happening here as they're pulled towards opposite sides. And then we have telophase. Telophase can be a little hard to see, I think, under a microscope. but what's happening is the nuclear membrane is starting to reform. And again, that's a little hard to see here, right? This is the entire cell. You can still sort of see some of those spindle fibers disappearing. The chromosomes start to unravel again. I think all these little dots are supposed to be the unraveling chromosomes, unraveling into what's called chromatin. Here you can see they're no longer the nice chromosomes. They look kind of bunchy and unravelly. And the cytoplasm starts to divide and cytokinesis begins. So one thing I like is you can really see here that there's other organelles, right? We focus so much on chromosomes during mitosis that we forget that there's organelles, other organelles there. But here's some mitochondria, here's some vacuoles, this looks like some endoplasmic reticulum, and so those are all of the other organelles. But really telophase is kind of the last stage where it's going to be one cell. And so then cytokinesis, which is not really considered part of mitosis. You remember from the first slide it said mitosis and cytokinesis is where the cells divide into daughter cells. And if you want to think of it as where the cells divide into two, that's fine. The term we use in science is daughter cells. The vocab, I say it again, not really concerned about you understanding the vocabulary of it. But what happens is the cellular membrane starts to pinch inward and eventually will connect here and here, and you'll get two identical cells. Again, think conservation of mass. Those two cells, if we go back to here, are clearly half the size of the one cell, right? And so that's why then it needs to go into G1 again and start to grow. And so remember back to all that carbon time stuff when it said like the mass comes from the girl itself, when it's talking about a growing girl or something. And some of you would say, well, yes, because the cells divide and that's where the mass comes from. That can't be where the mass comes from, because two cells that came from one are going to be smaller. Each cell is going to be smaller than the original for a conservation of mass. So that mass has still got to come from outside. It's still got to come from the food we eat. And then here you can see, it's a little hard to see under a microscope because it's hard to tell what's a cell before mitosis versus a cell after mitosis. But the way you can often tell is when it looks like two cells taking up the same space as like another cell next to it. Then you say, oh, I bet those have just undergone cytokinesis. So here are some further resources. I really care that you get big picture of cells growing, DNA replicating, and then again I'll show you kind of a hand device in class where you can kind of then see what I like to call the dance of the chromosomes. But if you're interested, you can look at crash course number 12, which is splitting up is hard to do, and that will give you much more detail on what's going on at each stage. Please feel free to put questions in the comment section. Have a good night.